Dear students, you can see on the screen the name of the website where you can find all the experiments. All the experiments you can find here. Okay, and this website address also I will write in the chat box so that you can uh, download all the experiments from here. You can find it with your leader also. So please, whenever you want any experiment, you can download it from here. Today we will perform two experiments. First experiment name you can see on the screen. It is laboratory safety. This is our first experiment. Dear students, you have done this experiment in your biochemistry also, so there should be no problem and it should be a very easy session. Today we will try to understand what is general safety rules. We have to identify the safety signs, and in the last we will describe the first aid process. But dear students, the most important thing, when you come in the lab, you have to be very careful. Yani A, B, C. Always be careful. There are many chemicals in the lab. There are many sharp objects in the lab. There are many acids in the lab. There are many electrical machines, appliances in the lab. So if you play around in the lab, then you can hurt yourself and you can hurt others also. So for your own safety, please be careful while you are in the lab, any lab. The most important thing you should remember is lab coat. You should not forget to wear a lab coat. Lab coat is a must in the lab because there are many chemicals and acids, and these can drop over your clothes, and your clothes can be dirty. So it is very important. When you come in the lab, as I told you, this is not a playground. So if you have any problem, just raise your hand, and I will ask the question, and I will try to answer all of your questions, whatever arise. When we deal with the general safety rule, the most important thing you should remember is if any chemical or acid drops over the table, don't do anything, don't play with it. As I told you, they are acid, they are chemicals, they are very dangerous, they can hurt you. So if this happens, just tell your teacher that this is the problem, so I will try to solve the problem myself. So this is very important to remember. If you wear glasses, Nazara, please do not wear contact lenses. Contact lenses, no. You can wear glasses. So you can wear glasses, okay, but do not wear contact lenses. In the lab, in the lab, we have fire extinguisher. We have fire extinguisher in the lab, okay. So if there is a fire in the lab, we have fire extinguisher to use. Also, we have fire blankets. We have a sand basket. So we have lots of things to fight with the fire. But inshallah ta'ala, it won't happen. But we have covered everything. As far as the glassware is concerned, uh, when you are working in the lab and you find some test tube or beaker or cylinder broken or cracked, don't do anything. Just tell the teacher, uh, and I will provide you with a new glassware, and you throw this glassware in a dustbin, in a bucket which is specific for glassware. So you do not throw it anywhere else. There are specific special buckets uh, to throw away the glasswares. So we have fire blankets, we have fire extinguisher, but very important thing you should remember, dear student, this slide is very important because we can ask in the exam what is the method of using fire extinguisher. So you should remember pass, PASS. P yani you will pull the pin. A means you will aim at the fire, base of the fire yani. Then you will squeeze. What is the meaning of squeeze? Press. And then you will cover the fire all around uh, with this. So this is the method when there is a fire in the lab. But if somebody gets fire, then what you will do? If somebody gets fire, what you will do? You will stop, drop, and roll. Stop, drop, and roll. What is the meaning? What is the meaning of this? It means that if you get a fire, first of all, stop running. Don't do anything. Second, drop yourself on the floor and cover your face. And number three, roll over the floor so that the fire is finished. So this is very important. But inshallah ta'ala, this kind of accident or incident will not happen in the lab, inshallah. After you finish your work, the most important thing is to clean the place, only your place. I am not talking about the whole lab, but your place, your table, your place where you're working, you need to clean it. All the slides and test tubes you need to clean, because alatul next group is coming and they don't know what you have done. So this can hurt others. So for your safety and for others, you need to clean your table before you go. Very important thing is when you are using microscope. Very important thing is to cover the microscope and close the microscope. When you finish it, close the microscope, turn it off, and cover it. So this is very very important thing to remember. As I told you, lab coat and gloves are very important in the lab. So you should know how to put on the gloves and how to remove the gloves. Dear students, as you know, corona is around and everybody knows how to use gloves. So this is just a few pictures just to show you how you can remove the gloves. Okay, so this is the method of removing of the gloves. No, we have gloves in the lab. So you just come, no need to bring anything, just bring your mask. You will come in the lab with your mask. Come on. Now also this slide you can see here, the method of washing of hands is shown here also. Because of the corona covid, everybody knows how to wash the hands and how much time, 20 to 40 seconds you should spend to clean, to wash your hands. So these few slides, they are telling you about washing the hands. Now if you are using needles, you are using needles or syringes, ibra. Okay, when you finish using it, you have to properly dispose of. You know why? Because there are some diseases like HIV, like hepatitis, HIV, which can spread through the syringes. So you need to throw them out properly in a special basket specific for which is specific for this. Okay, so all the syringes, everything, sharp objects, you will put in this basket and not in this basket. This is not for sharp objects. So uh, you should know how to dispose of sharp objects. Now, dear students, I am going to show you some of the pictures, and these pictures are very, very important from the examination point of view, because in the exam I can give you any picture and I can ask for the name of that picture. I think you have seen these pictures many times in your university, many times in the hospitals. So most of the pictures you already know. Uh, this slide, what you are seeing at the moment, students, this is very, very important. This is very important because I can give any picture and I can ask for the name. Okay, so do you recognize the picture? Do you recognize the picture? Okay, starting from here, starting from first picture, this is caution or warning. Caution or warning. Second picture, this is biohazard. Biohazard. This picture is biohazard. Then it is poison or toxic. Then it is flammable. Flammable. Then it is harmful or irritant. Harmful or irritant. And in the last we have corrosive for acid. So dear student, you should remember the picture and the name, and any picture can come in the exam and we can ask you to write down the name of this picture. So these are some of the signs you should remember for the exam, final exam. Okay, now this is a revision question. What objects you will throw here in this basket? As I told you, sharp objects, needles, and lancets you will throw in this bucket, not in the normal bucket. First aid. First aid. We have first aid boxes in every lab. We have first aid boxes in every lab. So if some problem occurs, you should not worry. First of all, the problem will not be a big problem. Second thing is we have this first aid box that has all the medicines, all the bandages. So don't worry about anything. Inshallah ta'ala, nothing big will happen. So we should use the first aid box if we get burn or cuts or bruises also. We have a first aid box wherever we are working, so that we can use it to manage. If somebody faints, you know the meaning of fainting? Fainting means unconscious. If somebody becomes unconscious, so don't worry, just put his head down and his foot up. His head down and his foot up. There will be a flow of blood towards his brain, and after some time he will become okay. There are many electrical appliances, so be very, very careful. But if somebody gets electric shock, don't worry, just turn off the main power supply, and inshallah ta'ala it will be good. Now dear students, this finishes my first experiment. Did you understand the experiment? If you want to ask any question, you can ask, otherwise I will proceed to next experiment. This is our second experiment. You can see on the screen. This experiment is about light microscope. Micro means very, very small thing, very small, so that you cannot see it using your eyes. So you need a machine, you need help. So this is the machine that will zoom the object so that you can see it properly. The objectives of today's experiment you can see on the screen. First of all, we will describe different types of the microscope. What are different types of microscope we have? Then we will describe different types of different parts of microscope. Okay, these are two main targets of today's experiment. As I told you, microscope we use for the objects that cannot be seen through our naked eye, so we use a microscope. There are three main types of microscope we have. Number one is the compound light microscope. Compound light microscope. Other types are electron microscope and fluorescent microscope. Electron microscope and fluorescent microscope. Dear students, we are using in the lab only compound microscope. We are not using electron microscope and fluorescent microscope. These microscopes are used in the research lab. IBHAS we use it. Okay. Now this is the picture of the compound light microscope. In the lab you will see this microscope. This microscope you will see in the lab. Okay. This microscope has two eyes. One eye, two eye, where you will put your eyes. That is why we call it binocular. This one has only one eye, so we call it monoocular. Mono means wahid, one. The other type of microscope is, you can see on the screen, it is electron microscope. Electron microscope is very powerful and it is used to see very very small objects like viruses, like bacteria, that cannot be seen using the compound light microscope. But we don't have this, and we don't have this also in the lab. Fluorescent microscope also we don't have. They are present in the research lab. So as I told you, uh, the main function of the microscope is magnification, takbir. This is the main function of the microscope, takbir, zoom. Okay, so when we talk about zoom, it means that microscope contains some lenses, adasa. So in the microscope we have two types of lenses. Yani they are muhaddib adasa, they are convex lenses, but there are two types. One is this, one is this one. This is called eyepiece or ocular lens, and second is this one, which is called objective lens. So we have two lenses. This is objective lens. Okay, objective. Okay, so these are two lenses we have in the microscope. Okay, the difference between these two, both of them convex. The difference between these two is that ocular lens قوة تكبير fixed, غير متغيرة, you cannot change it. It is 10 times, 10 مرات بس, you cannot change it. But objective lenses, they are more than one. Okay, أربعة حبة, four a power of قوة تكبير, 4 power of magnification. So if we want to change the power from small to big or big, we can change. This is the difference between the ocular lens and objective lens. So This is very important to understand. So see in this picture, ocular lens. I have told you ocular lens also called as eyepiece. But important thing is that the quwwa takbir fixed, quwwa takbir fixed 10x. On the other hand, objective lenses, there are four types: 4x, 10x, 40x, and 100x. It means that if we want a small zoom, we use 4x. If we want more zoom, then 10x. If we want more zoom, then 40 and 100x. So this is how it goes. This is the magnification power. This is the magnification power of the objective lens. But if you talk about total magnification power, yani there are two things: magnification power and total magnification, yani quwwa takbir ajmali, total magnification power. So how do you calculate total magnification power? When you multiply the magnification of the ocular lens and the magnification of the objective lens, you get total magnification. For example, if we are talking about 4x lens, 4 multiplied by 10, 40 is the total magnification. For 10x, 10 is the magnification. قوة تكبير. قوة تكبير إجمالي 100. قوة تكبير 40. قوة تكبير إجمالي 400. قوة تكبير 100. قوة تكبير إجمالي 1000. So you should understand the difference between magnification and total magnification. In the lab, we will be using uh, this lens paper. What is lens paper? Lens paper is a special type of mandil to clean the lens. So when we are using the microscope to clean the lens, we will not use any other tissue paper. There is a special type of tissue paper, otherwise the lens can damage. There can be scratches on the lens. To save the lens, we use this special kind of paper. Now dear students, this slide is very important, because many questions can come in the exam from this slide. This slide, this slide, this slide, this slide, and this slide, all of these slides, they are showing different parts of the microscope, and their function. So in the exam, we can ask about any number, what is the name, what is the function, you should remember. Now I am telling you all the names and the function. When you will come in the lab, inshallah, I will show you again the same things, and I will tell you the function again. Okay. First is first is number 1 and number 3. This I told you just now. I told you these are two lenses. These are two lenses: ocular lens and objective lens. So where is ocular lens? This is ocular lens. And where is objective lens? This is objective lens. This is objective lens. Okay, this is objective lens. What is the function of these? The function is magnification. As I told you, the function is magnification. Zoom, takbir. The only difference is that ocular lens only one power, 10. Objective lens 4. In this picture, you are seeing two lenses. Behind this, there are two more. So there are total 4 objective lenses. Okay. Now number 2. Number 2. Nose piece. Nose piece. Where is nose piece? This is nose piece. This is nose piece. Nose piece. Nose piece. Can you see nose piece? What is the function of nose piece? The function of nose piece is objective lenses are attached on the nose piece. So if you want to change quwwat takbir, you will use nose piece. You will move the nose piece and the objective lens will move. Yani from 4 to 10 to 40 to 100, when you are moving, you will use nose piece. Understand? Now, number 4 and number 5. Number 4 and number 5. Where is number 4? This is number 4 and number 5. Number 4, number 5. Can you see? These are two buttons. Number 4 button and number 5 button. What are these buttons? These are focusing button, focusing knob. Yani when you are focusing the slide, tadi, you will use these two buttons. When you will move these buttons, the slide will move up and down, fok tahat, so that you can focus the slide properly and you can see the slide clearly. Understand, students? Now number 6. Number, yes, number 6. Number 6 is stage. This is number 6. This is number 6. What is the function of number 6? Function of number 6 is you put slide here. It is the place where you will put the slide. Stage. Stage. Number 7. Number 7 is lamp. Lamp, yani lamba. Where is lamp? This is lamp. This is lamp. Can you find lamp? What is the function of lamp? Noor. When you need microscope, you need a light, okay, to make the slide bright. So this light will come from down, from this lamp. Now number 8. Number 8, condenser. What is condenser? Here you can find condenser. This is condenser. This is condenser. Can you find condenser here? This is condenser. Just below the stage. This is condenser. What is the function? The difference between number 4 and number 5 is that both of them, they will move the slide up and down, but number 4 is coarse. Yani coarse, yani when you will use number 4, the slide will move up and down fast. Number 5 is fine. When you will move number 5, the slide will move up and down but small. Okay, so we have special uses for number 4 and number 5. This I will tell you when you will come in the lab. I will explain what is the use of number 4 and what is the use of number 5. But the same thing is that both of them, they are focusing on. Now number 8, condenser. Condenser. What is condenser? Condenser is another lens. I told you two lenses, no? Ocular lens and no problem. Ocular lens and objective lens. This is third type of lens, but the function of condenser is not takbir. The function of condenser is tajmi'. Tajmi' an-nur. Yani when light will come from down, light will come from lamba, it will pass through the condenser, okay, then it will pass through the slide, then it will pass through the objective lens, then it will pass through the eyepiece or ocular lens, and you will see it. So condenser is a lens, but the function of condenser is not takbir. The function of condenser is taksir. Okay, tarkiz. uh, so I think you have understood. Now number 9. Number 9. This is number 9. Say this. This is number 9. Can you see number 9? It is mechanical stage. So it is present upon the stage. This is the mastara. This is the ruler present upon the stage, and this ruler is used to move the slide side by side. You remember number 4, number 5? They will move the slide up, down. But if you want to move the slide right, left, forward, backward, okay, so you use number 9, mechanical stage. mechanical stage is actually the ruler which is present upon the stage. And how you can use this ruler? You can use this ruler by this tahakkum. Number 10 is this tahakkum, this regulator, this controller. This controller, when you will use this controller, the slide will move side by side or forward backward. Do you understand, dear students? This is not focusing knob. This is to move the slide side by side. Now, next one is clip. Clip is, this one is the clip, this is the clip, okay, and this clip is used to hold the slide. When you will put the slide, this clip will hold it. So it is easy, I think. Now next thing is diaphragm. Diaphragm. Where is diaphragm? This is diaphragm. Just above the condenser we have diaphragm. What is the function of diaphragm? Diaphragm control the light. What is the meaning? The meaning is that sometimes you want eine fatih, bright. Sometimes you want eine dark. So this will control the light. Sometimes you want more light, sometimes you want less light. So this controls the amount of light that will come in the slide. Okay, sir. Now number 13. Now number 13. Number 13 is body tube. This is number 13. Can you see number 13? What is the function number 13? Very easy. Objective lenses are attached here. Body tube. Objective lenses are attached at a body tube. Now number 14. Number 14 is arm. Where is arm? This is arm. This arm is used to hold the microscope. When you move the microscope, you hold the microscope, you will hold it from here. And last thing is base. Ada. This is the base. Okay, no problem. 15 number is the base. You understand base? Now I will repeat number 13. This is number 13. This is number 13. Okay. And what is the function of this? This is body tube. What is the function of this number 13? Ocular lenses are attached here. Ocular lenses, eyepiece is attached upon body tube. I am repeating, ocular lens is attached in the body tube. Okay, so this is the function. I hope this answers your question. So next slides, they are just what I just told you. They are written here, so you can find it. This slide you can see how to hold the microscope. So this is yad, this is hand, this is arm to hold the microscope. The correct method of holding the microscope is not with one hand, it is with two hands. Otherwise it can damage the microscope. Now in the next slides, the method of using of the microscope is given. This you will understand when you will come in the lab. So I will tell you everything when you will come in the lab. Okay, when you will see the microscope, you will understand everything. But you have to remember that whenever you will put any specimen in the microscope, always you will start from 4x. Always start from 4x. Okay, 4x is called scanning lens. You know scanner? Scanning lens. Scanner means it will help you in the location of the phenyl. Okay, so when you will put shariha, you need to locate the specimen. So this you will do with 4. So always start from 4x. When you find the specimen, bring it in the center of the in the center of the vision. Then you can increase to 10x, which is low power. Then you can come to 40x, which is medium power, and then you will come to 100x, which is high power. But as you can see here, dear students, when you are using 100x, only 100x, when you are using 100x, you have to use a drop of oil, because in 100x the lens is too close to the slide. So this oil gives you a medium of refraction, inkisar. Okay, so you use a drop of oil only in 100x, not in any other. So you start from 4x, go to 10x, go to 40x alatul, but before going to 100x, put one drop of oil. This oil is a special oil, not cooking oil. So it's this oil I will give you whenever we will use 100x, but most of the time we will be using till 40x. Now, dear students, when you finish your experiment, you need to clean the place, you need to clean the slides, you need to close the microscope, you need to cover the microscope. Okay, okay, clean the microscope and cover the microscope is very important. As I told you, if you use 100x, you are using a drop of oil. So when you finish using the drop of oil, you need to clean it with the lens paper. So now in this experiment, first of all, in this video, first of all, you will see the microscope, different parts of the microscope that we discussed last time. And the other thing you will see here is the wet mount, how it makes the wet mount. Okay, so this person is now, this is the microscope he took from the cupboard. You know that he took this microscope with two hand, yani I told you last time, whenever you hold the microscope, always hold with two hands. Now he take it to the table. Okay, and now he just he is just telling you the function of the microscope is just like a adasa, just like a muhaddib adasa, just like convex lens that make us magnify the objects. If the objects are very very small, this can not work also, so we need a machine. So microscope is a machine. This machine has got two lenses, two muhaddibatsa. One is this, what he is showing, called as ocular lens. I showed you last time. Ocular lens, also called eyepiece. So this is one lens that has got a fixed quwwa takbir, which is 10, ashara marrat. Bas, we cannot change it. This is one type of lens. That second type of lens is this lens. Okay, see, he is now moving the lens. So these are the lenses, these are four lenses. These lenses are called objective lenses. Objective lens is another lens. Okay, and these lenses, they are different قوة تكبير, يعني ضرب أربعة, ضرب عشرة, ضرب أربعين, ضرب مية. So four objective lenses we have. So you see four objective lenses, and can you see how he is moving the objective lenses? He is moving the objective lenses by nose piece. When he is moving the nose piece, he is moving the objective lenses. So sometime you want 4x, you move it. Then you want 10x, you move it. So like this, you move the objective lenses. Okay, now he will show you the light. This is light. And now what he is showing you now, see this part is called diaphragm. What is the function of diaphragm? It controls the light in the microscope. Sometimes you want more light, sometimes you want less light. So that is how you control the light. This is also I told you last time. Okay, this part is also same. So forget about this. Okay, now this is stage. This is stage. This is the place where you put the slide. This is where you put the slide. And this part is a mastara. I told you this mastara is called mechanical stage. So what you do, you put the slide on the stage, you put the slide upon the stage and hold it with this clip, and then you move the slide right, left, forward, backward. ما في فوق تحت بس جهتين. You can move to sides, okay, by using mechanical stage. And how you use it? How you move the mechanical stage? Here. This part is called mechanical stage controller. Tahakkum. So when you move it, it can move forward, backward, and right, left. I think you understand the movement of the mechanical stage. Mechanical stage is moved right, left, forward, backward by moving mechanical stage controller. Okay. Now another thing is focusing knob. I told you focusing knob. These two focusing knob, they are important to move the slide up and down, to focus the slide. Tawdi. Okay. To make the slide clear. Big button. This button is called coarse focusing knob. This button is called coarse focusing knob. When you move this knob, the stage will move up and down fast. When you move a small button, fine focusing knob, stage will move slowly. Okay, so see, this is the fine focusing knob. Sometimes we use fine focusing knob, sometimes we use coarse focusing knob. So see, when he is moving coarse, see the stage is moving. See, stage is moving fast. Stage is moving fast. When he is using a small button, stage is using, but you are not seeing because the movement on the stage is slow. Now this is the lens paper. I told you about lens paper. This lens paper we use to clean the microscopic lens. Okay, so whenever you want to clean the lens, you use this manadil. You cannot use any other mandil. Okay,